have Dr. Taragan Ramji as a resource person. So I would like to invite Ms. Simi Kurian, Assistant Professor, School of Biosciences, MacFirst, to introduce the speaker.
Good afternoon to one and all. Today is the 8th day of our webinar series. It's a great pleasure for me to introduce our today's speaker. Dr. Taragan Ramji Moturu is a famous research scientist at University Liberty Brussels, Belgium. He did his doctorate in plant biology and bioinformatics, focusing on evolutionary aspects on stegolactone hormone and their role in stress adaptations. He has been awarded Wallonia Brussels International Grant to conduct his research work at University Liberty Brussels. During his 8 years of research experience, he published multiple works in reputed journals. He is truly excited about the applications of genomics and bioinformatics to disrupt the fields of agriculture and crop production. Presently, he is working on Arabidopsis, specifically natural variations governing the root morphological adaptation in response to nitrogen. He is a trained biologist with expertise in genomics and bioinformatics. He will enlighten us in his interactive talk about genomic-based precursor breeding approaches for improving plant stress tolerance. It's my pleasure to welcome you, sir. Over to you, sir.
Thank you. Thank you very much for your kind introduction. It's just, I just wanted to make sure it's audible and visible, right?
Yeah, it's audible, Raji.
Yes.
Audible. Yeah.
Thanks again for your introduction and also for inviting me for this current webinar series, current trends in the molecular life sciences. So I'll quickly share my presentation. Yeah. So yeah, thanks. So I will be mainly speaking about genomics-based precision breeding approaches for improving the stress tolerance. So as already mentioned in the introduction, so I'm a researcher here in a crop production biostimulation laboratory in University Libre, Brussels. So I got a WBI grant to conduct my research work here. So let's start with the keywords I used in my title. So first, what is stress? So as a biological stress is an adverse force or condition which inhibits the normal functioning and well-being of the biological system, such as plants, since we are talking about the plants. And it's then basically an organism's response to stresses such as environmental conditions. So it could be So it could be either abiotic stress or biotic stress. So abiotic stress could be like a heat, cold, drought, salt, metals, or any kind of flooding. Similarly, the biotic stress might include things like pathogens which try to attack the plants, or it could be insects, or it could be some random herbivores, or any microbes from the below ground which try to attack the plants. So the plants, since they are sessile, they develop a defense mechanism. So they have both developmental aspects, like using the primary metabolites to take care of the plants. So similarly, they have phytohormones to fight against this abiotic and biotic stress conditions as a defense mechanism. So let's come back to the current issues we have right now across the globe. So when you look at the projected impact of the climate change on the agriculture, on the climate change on the agriculture yields, so it was projected that there was a falling crop yields in the developing regions by 25%, and then like there will be a huge loss in the economy with it's almost like 84% of the economic impact of drought falls in the agriculture. And at the same time, the world population may surpass 10 billion by the end of the century. So this is like a common problem which you are already hearing about. So did you also know that so when you look at the percentage of our planet affected by the drought has more than doubled in last 40 years when you look at the 1977 projected to 2017. So it has been doubled in last 40 years because of this climate change and differences. So let's like stick to India itself. So when you look at the demographics, so 50% of India actually faces high to extreme water stress condition. So when you look at different areas, even now the present temperature has been risen more than the normal summer level temperatures. So this has been a drastic changes in the phenomenal changes you have been noticing because of the climate change and variations. So let's come back to the topic we are discussing. So what is plant breeding? So plant breeding is an art and a science of changing certain traits of plants over time in order to introduce the desired characteristics. So to improve the crop productivity, so that's our major target, to improve the crop productivity, to feed the overpopulating world, which we already discussed in the previous slide about when we talked about the issue about problems which we are facing right now. And so what are the different breeding strategies we have? So there are like multiple breeding strategies when you look at in a broad perspective. So there could be field-based testing, that is stress ecology, trait-based phenotyping, which is phenomics, or it could be gene or QTL-based screenings, which is part of the genomics, or it could be like a expression or interaction, which you're trying to check whether it could be environmental to the phenotype, or it could be a trait to the environment. So it could be different interactions. So with respect to the expression of changes inside the plants. So it could be either at the transcript level, which is transcriptomics, or at the protein level, it would be proteomics, or it could be at small metabolites, which will be a metabolomics, or it could be between two different things like or transcript proteome or transcript metabolite. So it could be an interactomics. So a different when you look at the interactions, it's a bigger network. So it's much better way to understand it, like how we can improve the productivity. So the other approaches are microbial approaches, which is microbiomes, in which it's more related to soil beneficial bacteria, which improve the growth of the plants and further boost the productivity. So one of the new frontiers which which improved during this time frame is CRISPR. I think which you already hear about. It's already been in the trials and testing. So this is one new frontier which has a huge potential in the present agriculture to improve the crop productivity and the yield. That's CRISPR-Cas based gene editing. So what is the conventional breeding technique which we have been using from decades? So it's basically like you have a simple virus. For example, I'll give you a simple start with a simple example. That is why you have a virus-resistant plant and a high-yielding crop plant. So you need a plant. You're looking for a plant which could be both viral resistant, and at the same time it has to have a high yield to improve the productivity. So you perform a cross and produce varieties and or induce mutations, natural mutations, select them, and then grow for the future generations. So that is the conventional breeding technique. So what's the problem with that? So time and precision. So it's extremely time consuming, and it's hard to be very precise. So that's why like as we improve in our technology, so new technologies has been arised to improve the current conventional breeding. So that is mainly genetic modifications. So where we will be able to specifically target. So what are the genes that are responsible for the for this resistance of for the resistance against this particular virus, and at the same time also the genes which are responsible for the high yield of this crop. So you mix them, you bring them together, you modify the genes, and then you develop this virus resistant and high crop yield, high yield crops. So, so when you look at the genome editing, so this is basically like target interventions at a molecular level of DNA delivered to alter the structure and functional characteristics of the organisms. So I mean, as you see in the picture, it's not, I mean, it looks simple as you cut the DNA and then bind it, but it's much more complex than as you think. So there are like different methods which are already developed. So one of them is it's a site-directed nucleases using site-directed nucleases enzymes, and other one is the CRISPR-Cas methods. So these are the common methods which are under use at the moment. So let's have a quick overview on zinc finger and talons. So zinc finger are basically nucleases enzymes, which nucleases are nothing but which cleave the DNA. So they have the DNA binding zinc finger motifs and an endonuclease fork1 enzyme. So this actually binds to it helps. So this basically motif helps to bind to the DNA, and this endonuclease fork1 actually cleave and make a cleavage in the DNA. So each module recognizes a nucleotide triplet, and then fork nuclear enzyme function as a dimer, and it cleaves it. So similarly, there are that's a second type of nucleases which are commonly used is talons. It's a DNA binding domain as well. It's amino acid repeats. It's as you see, it's an amino acid based repeats plus it's the same enzyme fork1. So this amino acid recognizes one nucleotide of the target DNA sequence, and this fork1 enzyme function as a dimer as before I was telling in the zinc finger nucleases, and make a cleavage and make a cut in the DNA. So the third one, which is much advanced, which is still in the infant stage, but it has a huge potential to change and to really edit the genome very precisely. So this is a genome by CRISPR-Cas system. So it has a single guided RNA bound to a nucleases. So which is this one. So it has a target specific sequence and other RNA sequence. So this binds to the Cas9 protein. So this Cas9 protein helps complex goes through the DNA until finding a match. So it looks for a match for the target sequence in the favorite gene which you want to cleave or cut them from the genome. And conformation change activates the nucleus. So when it starts to bind, there will be a conformational changes which activates the nuclease, and then this double-stranded DNA is cleaved and and then it will be repaired by the cell. So once after the cut, so you basically remove the gene. This is very target specific one. So where you cut and cleave based on the specific sequence. And then after making the cleave, cell has its own mechanism to repair the DNA back to double-stranded DNA. So what are the DNA? So you from the above from the last two slides, you have noticed like so we make a cut of the DNA, and then like a cell has its own mechanism to rebind to make the double-stranded DNA again. So so there are two different DNA repair mechanisms where genetic modification occurs. So this is basically a non-homologous end joining method where you produce a small insertion and deletion without the use of the exogenous DNA after cleaving. So, there is other way of repairing is the homology directed repair, where you can introduce a desired DNA sequence or gene in the targeted site. So after the cleaving, you can always have a donor template through which you can have a precise nucleotide alteration, or you can always have a insertion of the gene. So a mutation in the genome is induced either by this editing or deleting or inserting or replacing genes. So these are the different ways you alter the DNA, and then to improve or to put a desired character, or you want to remove unwanted phenotype from the plants. So again, coming back to the what are the drawbacks and what are the things which genome editing has? So genome editing to breed a better plants. Plant breeding has an enormous contribution to increase global food production, but there are some limitations. So the major limitation you always also been hearing is the GMOs, the genetically modified organisms. So basically we don't know so there are like pros and cons for this GMOs. So when you look at the pros, so it's a precise in inserting the desired trait. So you're able to insert the trait you're looking for, like a high yielding crop variety, or viral resistant, or herbivore resistant varieties. So you should be able to insert the desired trait you're looking for, and it's fast to obtain the final product since it's very precise and you know which trait you're targeting for. And you can increase the crop productivity and the quality. Example is producing resistant producing resistant plants, and fortifying with certain micro elements, etc., etc. So so when you look at the cons, like what are the issues? So when you so it's a random nature of gene insertion, and it might have an undesirable effects which we are not sure about, like in a so at one point you know that this trait has been inserted, but in a long run you're not sure whether this insertion is stable, or it may have a desirable or undesirable effects. So, and it's not favorable for making large concerted changes by changing the complete metabolic pathway. So it's only like a one gene or two genes which you could actually make changes in the in the plant, but not I mean, it's really laborious to make a change, to change a complete metabolic pathway. And also when you look at the policy side, it's completely heavily regulated because of multiple issues and uncertainties in this genome editing methods. So but genome editing to breed a better plant. So so again I mentioned, so this is the common method which by CRISPR-Cas method. So basically you isolate the protoplast, and then using the guided RNA you should be able to precisely target which gene you want to insert, or which gene you want to remove, and then further make a mutant or a mutant plant with the desired characters or desired traits. So this is very site-directed modifications you're trying to make from a new product. And by this also you're reducing the time to get a product. So when you look at the conventional methods, it takes a lot of time when you cross for generations, because usually the time frame or the life cycle of a wheat or rice, it's more than 6 months. So when you look at this, when you keep crossing, it will take at least a decade or 2 decades to at least to have one or two desired characters, desired traits. So whereas here in this case, in just one cycle, you'll be able to insert the desired character you're precisely looking for. So you're reducing the time of the getting the desired plants, and also you're reducing the number of plants involved. So when you look at the conventional methods, you have multiple varieties you need to screen, and further you're looking for the desired character or desired plant with the desired trait. So whereas here the screening process has been reduced drastically, as the precision is very as you are very precise in inserting the specific gene, and at the same time it's also possible to introduce a long DNA sequences as well. So it has really like a wide range of applications. So which could start with research. example, by elucidating a particular specific gene function by reverse genetics approach. And at the same time, you eliminate undesirable products that are negatively impacting the food quality or storage or processing. And you can accumulate a metabolite of specific value. You can fortify a specific metabolite in different plants, and you can simultaneously create mutations in a multiple gene family members. You can also create a variety of resistant plants to pests and diseases. Like, for example, this is well known, the rice resistant in Xanthomonas oryza. This is like a common disease in rice, which cause a drastic loss in the rice. So, as I said, there are like many others which could where this genome editing has an applications. So, so it's good. Like we have been looking at all these techniques that have been for introducing variations and stuff. So let's switch into more layman terms, or much in a very general outlook. So, food for a thought. Let's have this random thoughts. Do you know about the magnesium? So the magnesium is one of the micronutrient which are essential for all the living organisms, right? So magnesium is also essential for plants. So it's important in the chlorophyll, which is to have to perform the photosynthesis, and also important for the circadian clock in the plants. It's equally important for the also the impacts also on the human health. If you have known, if you have noticed your only dietary requirements. So 2/3 of the population actually do not meet the estimated average requirement for magnesium, and this is under recognized electrolyte disorder. So this is like not so much known in the, I mean, when we take the food. So this, when you look at, as I say, like when you look at the demographics again, the percentage of population meeting this recommended amount of magnesium is really pretty low. So and it has some effects, even though it's a micronutrient, it has some impact on the human health. So I mean, like it could be like a malnutrition symptoms, or like, I mean, the effects you could always see because of lower intake of the magnesium could be confusion, or depression, or irritability, or fatigue, or nostalgia you have. So this is called this. If you know the cartoon that related to the magnesium, it's called hypoglycemia 7. So these are like 7 different effects you can actually notice by hypo recommended level of the magnesium. So again, so how you can achieve the required amount of magnesium? So it could be a handful of cashews, or some bananas, 10 bananas, or 10 avocados, or this many amount of water bottles to require so this is 350 to 400 is the minimum requirement per day. So these are the different ways you could actually get it. But again, so how you can achieve this particular thing by having a equilibrated diet. If you know this five color diet, in which you eat 5 different colors of vegetables and fruits per day, to have a proper equilibrated diet. Similarly, either you can use you can have supplementation in the form of like a tablets or any other things, or you can have a fortified drinks which is rich in magnesium or any other micronutrient. Or can we also biofortify this specific one? It's a big question. It starts, so increasing the minerals or vitamins which are essential, and the humans are not able to achieve by the general diet. Can we biofortify it in the plants itself, so that by eating regularly, having a proper the normal diet, the humans could have the essential or the daily requirements. So that's one food for a thought. So this is one random example. I really was interested to show you guys. And so coming back, so I showed you the general perspective of the breeding process, and so like what are the essential, I have given one example of the essential element that is undertaken like with the present diet we have. So and so coming back again, I'll give you a one research example which we conduct in our present laboratory work. So so our lab, to give you a broad introduction, our lab is works on two different units. One is the crop nutrition unit, and other is a photobiocatalysis unit. So the upper unit focuses on the plants, specifically on the plants, where how we could improve the resources in the improve the resource use efficiency. And so again, it's connected with the other lab where we transform the waste and other things using the lytic polysaccharide monooxygenase enzymes, and then like it's connected again to the abiotic stress pathogens. So the two labs are very much interconnected in with respect to the plants and their transformation, their use with the other biostimulations. Sorry, yeah. So when you look at the plant nutrient scale, so plants perform photosynthesis, right? So from the light, so the use of carbon dioxide and water, the water is broken down in the chloroplast, and then it assimilates and produces the starch. So the major elements that are necessary for the plants include the nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. So the challenge we have taken upon was the challenge of sustainable nitrogen fertilization. So to decipher the mechanism underlying the nitrogen use efficiency in the plants. So when you look at this, so nitrogen use efficiency. So so the plants plants are sessile and they grow in the soil. So it could be the plant soil might be low in nitrogen, or it could be high in nitrogen. So the uptake, utilization, and remobilization, it's called the nitrogen use efficiency. So what happens? So when there is atmospheric nitrogen, that has to be converted into nitrate and nitrite forms for the plants to absorb, so that the root foraging occurs, and then the plants stay green and the productivity is high. Whereas in the other case, there is a low level of nitrogen because of low conversion of the atmospheric nitrogen to this one. The plants stay really small, and there is not much of crop productivity. So our laboratory strategy is basically from model to crop. So basically we look upon how does this nitrate influence the development, plant development, and the growth of the lateral roots. And then like we use the forward genetics approach and the natural variations, and this we transfer this knowledge from model to plant to the crop plant, like Brassica napus or oilseed rape. And we also work from lab to field so that we understand not just in the laboratory. We are not looking only at the laboratory scale, but also we are testing on the field scale, where we understand different variables, different climate variables that might be essential or influencing the nitrogen use efficiency as well. So I'll give you a quick one, the research, the work which we have been conducting at the moment. So it's basically we collected the Arabidopsis plants from their natural habitats, from our local regions in Brussels, from Netherlands, from a different country, in different areas. So we try to, so I'll quickly also show you, so we collected like from 210 sites, and so from every site we try to harvest at least 3 to 5 plants. And then so these are the different fields which we try to collect. So you can see when you look at the soil, it's completely different in different areas. So which we try to see like how it's actually affecting. So yeah, so when you look at like some of them in the French or the Netherlands area, so you can see the soil is a bit sandy, and it's completely different. The soil is it's a bit of dry, and we also collected some plants actually from close to the sea, which is more sandy, and it's the same plant again. So when we looked, so when we had a closer look, how these plants are actually varying. I'm sorry. How this plant phenotypes are varying in different natural habitats. So even though they belong to the same the same genus and species, still they have phenotypic changes based on they're adapted to the naturally to a different natural environmental niche. So you can clearly see a drastic differences between their root phenotypes from a different areas. So basically, these accessions represent from a different areas which we collected. So you could clearly see the length of the roots and the lateral roots are completely different because of their natural habitats. So we try to quantify them and measure them. So what are I'm trying to give you just the parameters which we looked upon, which are basically the root biomass, like how much does it weigh, or it could be shoot biomass, like how much the shoot weigh after like a certain amount of growth, and the total biomass and the root to shoot biomass ratios. And we closely had a look also on the root morphology, like how it's changing. So it's basically we divided them into like a zones, primary root, and we divided them into zone 2, where this is a primary where this is the primary root zone, and then the other two zones we divided for the complete primary root, and then number of lateral roots, so which emerge from site, and the sum of the what is the density of the lateral roots and length of the lateral roots. So these are the different morphological characters to see, to really quantify like what are the phenotypic changes they have adapted to during this changes in the natural conditions. So when we looked closely, so it was very clear the accessions are changing at low we try to grow them in a lower nitrogen level and a higher nitrogen level. So they were clearly made a distinct clusters. So where we could clearly see, okay, there is a distinct phenotypic changes based on their natural adaptation. They were able to grow in a different way when we grow them in a plates with the low amount of nitrogen and high amount of nitrogen. So, and also we divided them based on the characters of the different characteristics, as I mentioned previously, with respect to all the like the different traits which we try to use to understand their phenotypes. So what we do, so we had a different, now we have a detailed analysis of like how they are changing, how their phenotype looks like with respect to the shoots or the roots, and their growth in lower nitrogen, higher nitrogen. So basically from this, so we now we have to understand, so we are going back approach. So now you know the phenotype, right? So now you know the phenotype, like how it looks like. So now now we are interested, or we're keen to know like which gene or which are the like the pool of genes, one gene or multiple genes which are responsible for this specific trait or specific change in the phenotype. So we use the genomics approach basically here to really look into the like the changes like says at a nucleotide level. Let's say what are the mutations the plant naturally have made to adapt to such environment. So it's basically I'll try to simplify in a very layman terms. It's like it's a position at the different chromosomes, and so what you look at the dots, it's basically the SNPs, which with the so the one which is much above is with the highest likelihood of association with the phenotype of your interest. So you have a phenotype which has a longer route or a shorter route, or with a high density of lateral routes. So it could be anything, any difference, any different traits, or any drastic trait. So this SNP, which is like highly significant, could be the highest likelihood of association with this particular phenotype of interest. So we make this back approach, and then we score the SNPs, and then we look at the specific. So these are the. I mean, this is one example. Also, just I want to show you from the chromosome. So you look for different accessions, so different the plants which we collected from different fields. So you look at the SNPs, like and you score them, like, okay, this is the particular region that is responsible for this particular trait, and this is a particular SNP that is actually responsible to increase in the lateral root densities. So based on which we will be able to trace out, like, okay, these are the particular gene, particular mutations that are induced naturally to get that specific phenotype, and which helps in adapting to the lower nitrogen level of soil. So that's how we are transforming this specific knowledge where we identified this specific mutations, where mutations, and then we transfer this knowledge to the croplands to specifically precisely make that mutation to reduce the time which we usually take by the conventional breeding methods. So yeah, just to show you again, these are the fields which the fields in Belgium, where we grow the plants, where we transform the knowledge which we understand in the laboratory. So we try to also grow them in the fields, and we test them under different climate variables as well, to make sure, okay, whatever the mutation which we thought is interesting or inducing this particular trait is also like applied, and then we have a crop plant which is able to grow in a nitrogen deficit soil as well. So yeah, just to show you like how the fields look like, and this is what I mean. Like we have in the Belgium. This is oilseed rape, which we try to grow to understand at a different traits and different characters, phenotypes. So yeah, to conclude or to show the focus. So it's so our lab is basically a synergistic lab where and field experiments are employed to identify the specific genes and markers for this root morphology in response to the nitrogen supply, and also like we look for the introgression of this specific nitrogen root traits to improve the nitrogen use efficiency, and also this to remobilize this genotype could considerably limit the nitrogen pollution which is from the agriculture. And this root-based approaches for this ameliorating this nitrogen use efficiency is challenging, but definitely a promising process to improve the crop productivity. So yeah, thank you very much for your attention, and I would be happy to take any questions if you have.
Great session, sir. Now the section is open for discussion. Participants can ask directly your questions, or you can type in the chat box. Sir, I have one question, that is how CRISPR could transform our food supply. How far this helpful in our farmers? That means our farmers are facing lot of problems nowadays due to this abiotic stress. So can we make a sustainable agriculture?
Yeah, thank you for your question. So this is I would not disappoint you, but it's at the same time it's also like a, so as we have a technological advancement, it's important for us to really look back and forth, like if whatever the technology we are developing is well tested, and then we know its pros and cons before introducing it within the field. So that's the major, always the limitation or the bottleneck we always have. The best example which we are facing already is the COVID vaccine and stuff. So it has to, and similarly, it's also the CRISPR-Cas technology. It's still in the infant stages, where the scientists or researchers are still optimizing the methodology to make the precise insertion and deletion. So if we are able to achieve that precise insertion without any off targets and other stuff, and it has a really huge potential to really insert the desired traits. So I mean, as I mentioned in my presentation as well, so we could precisely put the traits which we are looking for, like for example, the high crop yield varieties, and also which are resistant to specific the infections which we commonly face in our fields, right? So India is largely dependent on agriculture. So this, I mean, it could really help to have a proper, and if it's a success, it has a huge potential to achieve that sustainable agriculture, as you mentioned as well.
Also, how this CRISPR affects the plant's defense mechanism. You already told about CRISPR technology, and plants have specialized defense mechanisms also there. How this CRISPR can affect plant's defense mechanism?
I'm sorry, I missed your question. It was broken. The last sentence was broken.
How this CRISPR affects plant's defense mechanism?
I'm sorry, I didn't get your question.
Hello, can you hear me?
Yeah, I can hear you, but so I mean, I didn't get your question.
How this CRISPR affect plants' defense mechanism? Okay.
So it's basically so you're trying. It's very targeted approach. The CRISPR genome editing is a very targeted approach where you precisely either make an insertion of a specific gene or a deletion of a gene, or you could insert specific mutation based on the desired traits and things which you're looking for. So, but the present, as I also said, like the present one has its limitations with respect to off targets. So it may also has so the deletion could be also have some deleterious effects on the other genes as well. So it's not still unclear. The methodology still has to be optimized for current use.
I think Sibasa has some questions.
So do I have to look at the chat, like so because it's really difficult for?
No. The CNMs will look through it and she will ask the questions. Read out the questions for you. Okay.
Okay. Yeah, it's really like difficult to.
Yeah, it's okay. Ramji, very nice presentation. Very simple and crispy. Very nice presentation.
Mm-hmm.
So you collected different ecotypes for conducting jivas from different locations.
Yes.
So you collected these ecotypes very randomly, or did you make some selection based on the soil nature or soil nitrogen content? Did you make any selection like that, or you just collected very randomly?
So the collection was made. So considering both, like it's also a random one. At the same time, also we collected the soil samples as well to make sure like we have distinct soil types. So like a sand, clay, silt. So we divided them into 3 different types of soils. So like so the ecotypes and the type of soil they are growing are completely different.
Okay, but anyway, yeah, you are making a correlation. Then once you analyze the phenotype, then you are making a correlation with the soil nature. Actually, are you doing that also?
Yeah. So that's also a part of one of the part of the projects as well. So to understand. Yeah. So it's the soil nature and how it's actually affecting the phenotype as well. So one is the nitrogen level, one is the soil nature to the phenotype. So basically the phenotype to all the other possible trait different models. Also with respect to the climate, because the climate in Brussels is different from the climate in the Netherlands as well. So slightly different. So we are trying to model this different variables that could influence the phenotype.
Okay. And how far you are with this Jiva study? I mean, you already have your candidates. How many candidates you picked already from your Jivas? So it's already or still going on, or it's already finished?
So we have the GWAS data already. So the analysis is actually in progress. So with respect to the phenotype and other things, we already have the data.
Okay.
So we are trying to pick up the genes at the moment.
Okay, Ranji. Thank you.
We have a couple of questions in our chat box. Deepthi Suresh Kumar is asking about, can we use genomics-based breeding to increase crop yield under drought stress?
Yeah, definitely. Yeah, definitely. So it's first, the important part would be first to understand really. So that's where the basic sciences come into picture. So first we need to understand like what are the genes or what are the like the traits or specific QTLs which are responsible for that inducing the drought resistance. So once we have that picture, clear picture, we can always transform this knowledge to the plants or by inducing the mutation or by inserting specific genes and structure. So it's first important to understand or have a clear picture, like so what are the QTLs or what are the genes that are responsible for this.
Another question is from Vijida B. Nair. Does genomic selection have a future in plant breeding?
Come again? Sorry,
Does genomic selection have a future in plant breeding?
Yeah, definitely. So right now the plant breeding has been advanced because of the genomics. So the best example I have given you is from my research work itself. So I have been using the genomics-based approach, GWAS studies. So we sequence the genome and we try to look for the SNPs, the single nucleotide polymorphisms that are responsible for these traits or the phenotypes. So this has been accelerated. The genomics, because of this genomics technologies, there has been acceleration in the breeding process compared to the conventional one where the farmer looks for the phenotype and then they try to get together them in the next generation and next generation. So it's really a laborious process when you look at the conventional one. So when you look at the genomics way, so you know like which plants have the specific traits. So it's easy to cross and then to develop to bring that specific character into the next generations.
The next question is about whether any particular criteria for a plant or animal to be transformed into a genetically modified organism.
So the answer would be simple. It's just so when you say the abbreviation itself has the answer. So it's when you say it's genetically modified. So whenever you try to introduce something foreign other than the natural one, it's genetically modified. So you're basically inserting, removing, or trying to replace a single nucleotide with something. So it's a genetically modified.
Dr. Salim Jose is asking whether it is fit for Indian farmers.
I'm sorry?
Whether it is fit for Indian farmers.
So I mean, I was also working previously with the wheat. So I was working with wheat where we had fields in Pusa, Bihar, and Rajasthan. So we had also. So this is one example I could give from my previous experience. It's so we had which is one crop with high productivity and one crop with which is resistant to spot blotch infection. So we tried to cross them so that we could have that hybrid variety where the crop has both spot blotch, the resistance, and at the same time with higher productivity. So using this technology, we were able to accelerate the process, and then we were able to come up with the inbreed lines, so which are actually carrying the both traits with a high productivity and also resistant to this particular spot blotch infection. So that's so I mean so once we so since we did not introduce anything foreign, so it's a natural variety which we developed using this genomics approach, and it was actually also used by the farmers as well. So it's a matter of whether it's developed by a traditional breeding technique or or a GMO. So GMO has its own regulations, because of which we are not able to use it directly directly for the farmers. But whereas using the breeding techniques, we were able to the farmers are able to actually use it when it comes to also Indian farmers.
Okay, another question is from Sumira Rafi. How much time do you think this technology will be available to a farmer in India?
Well, it depends. So I could say there are two bottlenecks again. So one is the advancement of the technology. So if once the procedure and protocol is optimized for the plants, for the insertion or any changes in the DNA, in the genome, that's one bottleneck you have. So once you have the technology, that's one. And the second one is at the policy level. So the government regulations. So the government needs to understand again, like the GMOs are not really doesn't have any health effects on the humans. So that's the second thing. Again, that's also a big bottleneck which we always have. So EU has been, like a European Union has been struggling to introduce the GMOs into the fields directly, and there has been a discussion between the scientists and the policymakers. So so it's completely depends on government to government, and it's kind of hard to really say like in this particular time frame we will be able to achieve it. So I mean, technology-wise, definitely we are accelerating at a good pace. But when it comes to the policy and implementation, it depends completely on different governments.
Next question, are disease resistance in crops and root exodus interrelated?
And root exodus, right?
Yeah.
So I mean, like it's two different aspects. So root exodus is basically I mean, like a small metabolites which are released by the roots just to attract the microbes to form that interaction, soil rhizosphere interactions. But also at the same time, this could also help. This plant rhizosphere interactions could also help in the plant defense mechanism as well. Like there has been a couple of reports where it could enhance the plant defense mechanism above shoot level with respect to getting resistant to the pests, insects, or herbivore attacks. So I don't say it's both are the same, but at the same time they're correlated or interrelated with respect to the interaction.
How can we extract root exodus in laboratory conditions?
So I mean, like either you could directly use in the plant medium. So it depends on the type of the plant which you're working on. So it could be directly introduced if you're working only on the seedling level, it could be directly introduced into the growth medium, MS medium, which you use. Or it could be also when you're growing them in a soil, it could be sprayed, or it could be in a different ways. So it completely depends also on the type of plant which you're working, and then type of way the plant growth ways. So it could be either at the plate or hydroponics, or it could be at the soil. So it has, based on which you actually optimize and decide like which is better for testing.
Okay. What are the breeding approaches for improving cadmium stress tolerance in plants?
Cadmium stress.
Yeah.
Specifically, I don't really know, but I never worked on cadmium. But yeah, I mean, like when you look at the stress, it's I mean, even though it could be a different one, but it's still like the responses are almost interregulated, or the similar way the plants show, which I already showed in the presentation as well.
Okay, another question is from Alfonso Thomas. She is asking whether gene-edited crop varieties are safe.
Yeah, this is quite a debatable question again. So it's kind of hard to say because so from the main problem which I also see is, so we know that, like, okay, when we produce a GMO, it's we are inducing some specific desired trait or gene through which there are no other effects on the plants, which also doesn't affect the humans. So this is in a short time frame which we notice, but we don't know long-term effects, like how it could potentially change or have influence on the humans. So this is something which that need to be intensively studied, and at the same time also, like how this artificially, so how this genetically modified, so how stable the variation which we introduced in the genome of the in the plants, so how stable it is. It's also on the other side, it's on the plant side, this is one aspect, and at the same time, like how it's influencing the human health on a long term. So this is the two different aspects which need to be studied. I mean, it's still not unclear. That's why it's really debatable whether we could use this generated GMOs into our normal food process, food as a food.
Another question is from Dr. Salim Jose. He is asking whether it is easy to get the seeds for propagation of crops every season.
So it depends again on the type of crop you're looking for. So I mean, like, there could be seasonal and perennial crops. So it's completely different. It depends on that. But I mean, when you grow them in a laboratory conditions, under controlled climatic conditions, you can always get the seed. You can propagate the seeds like in a controlled condition. So you can always have seeds when you want. But when you're growing them in a fields, it could be a seasonal or perennial crops. So depending on that.
Thank you so much, sir, for a nice presentation.
Thank you very much again, like, for the invitation, and I specifically want to thank Sibu for inviting me for this wonderful webinar series. And yeah, thank you.
Okay, Devika.
Yes, sir. Thank you, sir, for the knowledgeable lecture. Now, I would like to invite Ms. Serene Raju for proposing the vote of.
Okay, ma'am, am I audible?
Yes.
Good evening, everyone. I'm really gratified to deliver a word of thanks on this proud occasion of International Webinar Series. First and foremost, I would like to extend my sincere gratitude to our beloved principal, Reverend Father Dr. Chirian J. Kotel, for always supporting us and leading this institution in such a prestigious manner. Thank you, Father. The success of a webinar is immensely dependent on the speaker. I'm very grateful and I want to thank our speaker, Dr. Taraga Ramji Moturu, on behalf of the entire MacFirst family for enlightening all of us with a very informative and interactive session. Sir, your deep and intellectual way of imparting knowledge has added to the glory of this webinar. Thank you, sir, for your valuable time and effort. Now, I would like to thank our head of the department, School of Biosciences, Dr. Jenny Jacob, for always being a great motivation and taking constant initiatives to give us the best. Thank you, ma'am. Next, I take this opportunity to mention our deep sense of appreciation to our coordinator, Dr. Sibu Simon. Thank you, sir, for your great efforts in making this webinar a huge success. Now, I would like to extend my special gratitude to the Dean, School of Biosciences, Dr. Beena Cherian. Your presence and support has added to the glory of this event. Thank you, ma'am. Further, I take this opportunity to thank our research coordinator, Dr. Jodish Kumar, for his moral support and guidance. Thank you, sir. Finally, all the listeners who have made this event more graceful with their active participation, thank you for being with us for making this webinar a grand success. Wish you all a good health. Once again, thank you.
Okay, Ramji, thank you so much, Ramji. Yeah,
Thank
we
you.
will be in touch.
Thank
We'll be
you.
in touch. Yeah.
Thank you, sir, for accepting our invitation and coming here. Thank you.
Absolutely, my pleasure.
Okay.
So probably we can be in touch again.
Absolutely, no problem. Yeah, of course.
Yes.
Okay,
Thank you.
Ramji. Then bye.
Please fill the feedback link before you leave.